in section 9.6, we look at some aspects of whole muscle contraction. And the very first thing that we look at is the composition of muscles themselves in terms of the three types of muscle fibers that we previously discussed. Just to review, we have three types of muscle fibers. Slow oxidative fibers, which are known as type 1, fast oxidative glycolytic fibers, which are known as type 2, and fast glycolytic fibers, which are known as type 3. We can see all those fiber types illustrated down here in the lower portion of our screen in the figure from our text. And what this figure is showing us is that any muscle in the body is composed of motor units of each of these three fiber types. And so this is supposed to be a depiction of an actual muscle. And you can see that in this particular muscle, there are type 3, type 2, and type 1 fibers that make up the muscle. And we would expect to see that in any muscle that we look at in the body. However, depending on the function of the muscle, then we would expect a higher percentage of one fiber type or another. Take back muscles as an example. So a muscle that must remain contracted for long periods of time, allowing us to remain upright as we sit or stand. It's very important for those muscles not to fatigue easily, and therefore we would expect them to have a higher percentage of slow oxidative fibers, type 1 fibers, as opposed to type 2 or type 3 fibers. And that's exactly what we see. Or take our arms as an example. And so arms are not typically undergoing strenuous contractions unless we need to throw something or punch somebody. And if that's the case, then they need to generate a lot of force very quickly. And so it would make sense that they would possess a higher percentage of type 3 or fast glycolytic fibers, and that's exactly what we notice in our arms. It's also interesting to note that the type of fibers that a person possesses vary greatly from person to person. So if we look at the quadriceps of really great distance runners, and so these are not people that can run 3 miles, but these are people that do super well in marathons. And if we look at the percentage of the different fiber types that are found in their quadriceps, we see that really good distance runners have a very high percentage of slow twitch muscle fibers, whereas sprinters are nearly the opposite, having a very high percentage, as much as 75% of fast twitch, which would be type 2 and type 3 muscle fibers. And so interestingly, much of this is the result of genetics. This is why, as I've told you before, basketball never worked out that well for me in high school. Despite what Coach Williams told me, I couldn't actually achieve anything I set my mind to if I worked hard enough because of my genes. I simply have too many slow twitch muscle fibers in my quadriceps to ever be fast enough to really excel at basketball. But if you look at great athletes, then you see just the opposite of that. And so, of course, depending on the sport, but sports that require quickness, those people would have a super high percentage of fast twitch type 2 and type 3 muscle fibers located in their quadriceps and other parts of their body. So this also applies, interestingly, to animals. And so this takes us to turkeys. We often ask the question at this particular point, can turkeys fly? When I ask this question in class, I often get mixed answers. Some people are under the impression that turkeys cannot fly. So let's ask that question again. Can turkeys fly? Clearly, yes. Turkeys can fly. Which takes us back to our question regarding the muscles that are found in turkeys and the fiber types that are found in different parts of turkeys. So, as everyone knows, the best turkey meat is the dark meat. And so the dark meat, of course, represents muscles that are composed primarily of slow oxidative fibers, type 1 fibers. These fibers, as you would imagine, are found in the turkey's legs. And so the turkeys need to be walking around most of the time. They can fly, but they're not soaring overhead like vultures or hawks would be. So most of the time they're on the ground walking, and they have to maintain posture similar to what humans do. And so it makes sense that their legs contain a high percentage of motor units composed of slow oxidative fibers or type 1 fibers. However, if you look at the muscles that control their wings found in the breast of the turkey, then what we notice is that turkeys can fly, as we saw in that video, but as I mentioned, they're not cruising overhead like a hawk would be. And so those muscles that control the wings typically have to just exert energy over very short periods of time, similar to our arm muscles. And so it's in the breast muscle that we find primarily type 2 and especially type 3 fast glycolytic fiber motor units. And so that means that when we look at turkey meat, the muscle that comes from the breast appears white. Interestingly, the tension that's generated in a whole muscle depends on two things: the tension that's produced by each fiber that composes that muscle, and number two, the number of fibers that are contracting at any given time. Now, as we've discussed previously, fibers can only be recruited via a motor unit. And so a motor unit is either activated or not. There is no partial activation of a motor unit. And therefore, the number of fibers contracting depends on how many motor units are active in a particular muscle and how large each of those motor units are. Because, as we go down to the next bullet point here, we do notice, as we mentioned previously, that motor unit size varies dramatically in different muscles of the body. If we look at the muscles of our eye, the lateral rectus, medial rectus, and so forth, what we find here are some of the most finely tuned muscles of the body. We have the ability to undergo very slight contractions that move our eyes in very small to very small degrees in one direction or another. So in this case, we find some of the smallest motor units in the body. In other words, we have one motor neuron connected to as few as 13 muscle fibers. On the other hand, if we go down to our quadriceps, we find some of the largest motor units in the body. This is a muscle that doesn't need to undergo very fine degrees of contraction or doesn't require very precise control, but it needs to generate a lot of force in a very short period of time. So here we find huge motor units. We find one motor neuron connected to perhaps thousands of muscle fibers, and this allows us to contract this muscle very quickly or activate these muscle fibers very quickly, which allows very rapid contraction of this muscle, but not precise control, as we see in the muscles of the eye where we have very small motor units. Okay, a note here as we go down to our third bullet point regarding the tension produced by each fiber. And so this is just a reminder. Type 3 fibers have a greater diameter, as we remember from our previous discussion. And this means that if we have two motor units side by side, we have one that has 100 type 3 fibers, and we have another that has 100 type 1 fibers. They're both activated. Well, the one that has the type 3 fibers is going to generate more force because we simply have more myosin and actin filaments found in those larger diameter type 3 fibers. It's also worth noting that type 3 fibers tend to have larger motor units. This is not, of course, directly related to the fact that their fibers are larger, but it makes sense based on the fact that these fibers, the type 3 fibers, are usually involved in generating a lot of force in a short period of time, such as in the large muscles of our arm or the large muscles of our leg, and it's here we find very large motor units. Now, as we mentioned previously, we have to remember that a single muscle consists of motor units that are comprised of all three fiber types. Maybe I should restate that. If we look at a single muscle, we find all three, type 1, type 2, type 3 muscle types within that muscle. Now, of course, each motor unit only consists of type 1 or type 2 or type 3, but in a muscle, we do find all three of those fiber types. And interestingly, when a single muscle contracts, the recruitment of the different motor units that are composed of the different fiber types, type 1, type 2, and type 3, is different. All the different fiber types are not activated simultaneously. And so to look at this, let's go up to our diagram that's found on this particular slide, and what we see is the following. So what we notice is that if we apply a stimulus, then the very first, on the y-axis here we have whole muscle tension, on the x-axis we have time. And so the very first muscle fibers to respond are the type 1 fibers, or the slow oxidative fibers. These fibers respond most rapidly because the diameter of the motor neurons that are innervating these fibers are the smallest diameter. And so when this activation occurs in the central nervous system, we have excitatory impulses that are sent to all three of the cell bodies of these motor neurons. But the motor neurons of the type 3 fibers are the smallest, and so the same amount of stimulus affects them more. So we achieve action potentials most rapidly in these type 1 motor neurons that are going to activate these type 1 motor units consisting of the slow oxidative fibers. If we continue to send, in other words, if that little bit of contraction generated by the slow oxidative fibers is not sufficient, and we continue to send these excitatory impulses, then if we follow our diagram here to the right time-wise, we see that here the type 2 motor units are activated, and then when we get to about 40% of the maximal tension, which we'd have to look at here on the y-axis, only then are the type 3 fibers recruited. And so interestingly, even in a muscle like a bicep, which has a very high concentration of type 3 fibers, in other words, fast glycolytic fibers, then we do see that the type 1 fibers will be activated first when we're picking up a huge dumbbell. The type 2 fibers will be activated next, and then when we get to 40%, the type 3 fibers would be activated. And so to summarize, what we see here is that whole muscle contraction depends primarily on this process of recruitment that we just described. Now, the reason that I have this particular, if you look over here to the lower figure, this particular figure on this slide, again, it's the same figure we looked at previously, is to remind us that as a result of action potential frequency, we can vary the tension created by a muscle fiber only 3 to 5-fold. So this is showing about 3-fold. We can get up to 5-fold with an increased number of action potentials and reaching tetanus, but that's it. And so if we look at the muscles of the body, we can vary the strength of contraction vastly more than that. We can undergo very weak contractions or very powerful contractions with most of the muscles that are found in our body. And so nearly all of that is the result of recruitment. It depends on how many motor units are activated within a particular muscle fiber. If we activate a small number, we have weak contractions. If we innervate a large number of muscle motor units, then we can undergo very powerful contractions. What we notice is the following, that muscles are very sensitive to use and to disuse. And so if we start at the top of our slide, we do notice that disuse causes a decrease in the overall size of the muscle. So we can observe that through two types of atrophy. One is denervation atrophy, in which disturbingly the motor neurons to a muscle are actually destroyed or severed in some way, so the muscle cannot receive electrical stimulation, action potentials, in other words, to contract. And the second one is simply disuse atrophy. And so disuse atrophy is most typically associated with something like a broken arm and having a limb immobilized within a cast, and so it can't contract, at least to any great extent, for a certain period of time. In both of these cases, each individual muscle fiber becomes smaller as a result of the loss of myosin and actin filaments as we go through time. Conversely, what we notice, of course, is that use increases the number of muscle proteins that are present, and therefore the overall size, at least in many cases, of the muscle fibers. But interestingly, depending on the type of use that we're referring to, we have different results. And so aerobic exercise doesn't always increase the size of the muscles. In fact, as we see in our second bullet point here, the fiber diameter can even decrease slightly. But what we do see is that we have an increased number of mitochondria, an increased number of capillaries, and other systems like the respiratory system, the cardiovascular system are impacted, and they're impacted in a way that improves the ability to deliver oxygen to those muscles. On the other hand, what we notice is that strength training. So in other words, what Arnold's doing here in the top slide, strength training increases the number of myofibrils that are found in type 3 muscle fibers. And so it might be useful to go back and remind yourself, if you have any doubts on what a myofibril actually is. The myofibrils, of course, are the units of the myofibers. The myofibrils, interestingly, apparently add proteins in response to strength training, increase in size, and then split. So you can have more myofibrils, but you don't to any great extent generate a higher number of muscle fibers. Strength training also increases the number of glycolytic enzymes that are present, and it has neural effects, such as increasing the recruitment of type 3 fibers, making that more effective, and also, as we've talked about once before in class, reducing the input from these inhibitory sensory receptors that are responsive to stretch. And so when you pick up that huge dumbbell and undergo that curl, this stretch receptor tells you that there's a tremendous amount of force being applied to the muscle, and so there's this reflex that in turn activates these muscles that the antagonist muscle that will decrease the tension on that bicep. And so eventually strength training inhibits, to some extent at least, apparently, that reflex and allows a greater amount of weight to be lifted. But overall, what we see is that strength training then increases the number of type 3 fibers. And so the muscles that you see there in a weightlifter are the result of increase in size of those type 3 fibers. Those fibers, as we've learned about previously, are not resistant at all to fatigue, and so it's not going to help you in any sort of distance type of exercise. On the other hand, aerobic exercise, while it will help you in terms of the distance events, it has no impact on the overall size of your muscles. In fact, it may even decrease the size of the muscles ever so slightly. Now, sadly, what we notice is this, that you're not going to get a whole lot faster, or at least that's what our best science tells us. You might get a little bit faster, and so that was my problem, as I mentioned previously in high school basketball. So there's little change. Research indicates that there's very little change from slow type 1 fibers to fast type 2 and type 3 fibers, perhaps 10% or so, with really intense training, trying to increase your 40-yard dash time or something of that nature. It's not going to be very effective because those myosin heads are fixed as a result of your genetics, and there's no changing that. It is, evidence does indicate that you can change some of the type 2 fibers to type 3 fibers. In other words, the fast oxidative fibers to the fast glycolytic fibers. So you can get a little bit better at, or to some degree, you can improve your ability, your endurance ability, but it's going to be harder to make yourself a great deal faster as a result of training, exercise of any sort. Lastly, I'll mention that we have, in this particular picture, Scott Kelly and his brother. And so Scott Kelly is on this particular slide. I should have mentioned it earlier as a result of this first bullet, which is disuse and muscle atrophy. So Scott Kelly at the time was, a couple of years ago, the astronaut that spent the longest period of time in space, just over a year if I'm not mistaken. And so his twin brother is shown beside him in that picture. He interestingly had an identical twin And so, as you would imagine, that generated a great deal of interest in comparing the two when he returned from space. And of course, in space, we have no gravity, and so the muscles are experiencing some degree of atrophy, even with intentional exercises to, with bands of some sort to counteract that. All right, so what we see is that there are mechanisms by which muscles increase in size are incompletely understood, but the three things listed on the screen here certainly play some role. So first off, steroids. So androgens certainly play a role in muscle development. No question about that. Locally produced insulin-like growth factor also plays a large role in muscle development. But lastly, interestingly, hormone referred to as myostatin, interestingly, appears to play a role as well. And so myostatin is a hormone that works in a negative feedback loop to inhibit the overdevelopment of muscles. And so we can see the role of myostatin in a very pronounced manner by looking at Belgian blue cattle. And so this is a Belgian blue bull, this is a Belgian blue bull. Belgian blue bull and Belgian blue cattle, interestingly, are myostatin mutants. They are a breed that has existed for a number of years, hundreds of years at least. And of course, before the past few decades, when we had genetic testing and could sequence genes, we didn't know the reason for this. But now we can clearly see that they are homozygous myostatin mutants. They don't have myostatin to inhibit muscle development, and so they grow to these huge sizes with this massive muscle development. They're not extremely popular as a result of the fact that C-sections are often necessary for the calves to be born, but they certainly have remarkable muscle development. I remember seeing one of these at the state fair last August, and it was remarkable as you looked around. All these were super nice cattle, but these guys looked completely different. It looked like they had been on the treadmill and in the gym, and of course they hadn't, but simply it's this result of them being a myostatin mutant. The discovery of myostatin, as you would imagine, has generated a great deal of interest in developing a method to be able to control it. As we look at the screen, we notice on the left we have a control mouse, and on the right we have a myostatin inhibited mouse. And so there's a great deal of interest in developing myostatin inhibitors in order to help people who have muscle deterioration diseases, some of which we'll talk about momentarily. Of course, there are also the people who will take almost anything, and you see an example of that on the right. If you Google search this, you get all kinds of myostatin inhibitors. Hopefully most are just complete quackery, but it's possible that some would contain something that would actually be harmful. We also noticed that the ability of muscles to respond to exercise, disappointingly, decreases with age. And so we see that on average we have a maximum force decrease of 30 to 40% between the ages of 30 and 80. And so we see this decrease in muscle diameter that corresponds with that. But we do notice that this in part may be due to a lack in exercise, and so the muscles are still responsive to exercise, mostly, although unfortunately the degree to which they respond does decline to some extent as we go through time. Lastly, on this particular slide, we discussed briefly soreness, and so we're all aware of the fact that following exercise, or at least exercise to which we're not accustomed, we experience soreness the following day. And so this soreness appears to be the result of actual damage to muscle fiber proteins. And so in electron micrographs of muscles following exercise, we see broken thin filaments, twisted Z discs, and apparently in response to this we have histamine release, which results in the pain and soreness, and then the muscles rebuild themselves, and in order to counteract that damage, they rebuild in a manner that is somewhat stronger, at least in if you're, well, should be at any age, although it happens, as we mentioned above, more effectively in younger individuals. And so those muscles rebuild in a way to try to prevent that damage in the future. It's also interesting to note that eccentric, so lengthening, versus concentric, which are shortening contractions, tend to generate the highest degrees of soreness and the highest increase in strength. As we finish up section 9.6, we want to take just a moment to talk about some terms related to muscle contraction and the nature of muscle contraction. So we're probably aware of these terms, but we want to make sure that we do remember the terms flexion and extension. Flexion always refers to the decrease in joint angle during muscle contraction. And so when we flex our arm, we are measuring the angle between the upper arm bones and the lower arm bones. And so here we'd have 90 degrees, and then of course here we have extension. And so in extension we're increasing the joint angle, and then in flexion we're decreasing that joint angle. And that's true, we can apply that to any of the joints in our body. Muscles, of course, can only shorten, as you well know, and so one muscle at least, only has the ability to shorten and pull two bones closer together, but muscles are arranged into these antagonistic pairs. And in the particular case, like the arm, as an example, the bicep, of course, results in flexion, and the tricep results in extension. So muscles are always arranged into these opposing antagonistic pairs that allow muscles to function. One thing that we notice as we round out this little discussion is that oftentimes the action of a muscle depends on the contracted state of other muscles. And so, as an example, let's take a look at our gastrocnemius and our quadriceps. And so what we see is the following. If our quadriceps are relaxed and we contract our gastrocnemius, then we have flexion of the leg. And so this is something that occurs in the normal process of walking. And so that's the result of the contraction of the gastrocnemius in that particular situation. However, if we contract our quadriceps and we also contract our gastrocnemius, then the leg can no longer flex as a result of the contracted quadricep. And so the only thing that can happen is that the foot extends, as if we were standing on our tiptoes. And so interestingly, as we look across the body, we frequently see this, that the action that a particular muscle brings about depends on the contraction state of other muscles that are associated with nearby parts of the body. In section 9.7, we talk briefly about a couple different skeletal muscle disorders, and this will actually bring our discussion of skeletal muscle to a close. The first disorder we talk about is hypocalcemic tetany. And hypocalcemic tetany, interestingly, is a result of low calcium levels that are found outside the sarcolemma. This might not be an expected result given the role that calcium plays in muscle contraction, but what we notice is that the low calcium outside the cell has a direct role on the sodium channels that are found in the sarcolemma. And so it causes an increase in the opening of these sodium channels, an increased number of action potentials, and therefore these tetanic contractions that are not brought about by the central nervous system but are brought about locally as a result of these low levels of calcium. So the second disorder that we talk about is muscular dystrophy. And so muscular dystrophy occurs in 1 in 3,500 males. It's an X-linked trait. There are different mutations that cause it, and I should point out some are X-linked and some are not, but the most common cause is Duchenne muscular dystrophy, which is X-linked. Muscular dystrophy is not a problem with the muscle fibers themselves, so not with the myosin actin, troponin, tropomyosin, even the Z-discs or the M-line or anything of that nature, but rather the costomeres. And so the costomeres shown here are the proteins that connect the muscle fibers to the sarcolemma and therefore transfer the force from the contracting muscle fibers to the overall muscle cell itself. And so in muscular dystrophy, these are weakened, or like in Duchenne muscular dystrophy, there's a mutation in the protein dystrophin. Now that's where the name for muscular dystrophy came from. Dystrophin is an essential protein found in these costomeres, and as a result of this mutation, the costomeres, it's not that they're non-functional from the start, but they break down very easily. And so the symptoms typically show up in the first 6 years of life, and there's a 20-year life expectancy associated with Duchenne muscular dystrophy. Hopefully, although I haven't, our book doesn't address this given the recent nature of these events in science, but hopefully CRISPR will allow us to insert the correct genes and make real progress in correcting this particular disorder. Myasthenia gravis is a collection of autoimmune disorders that are actually more common in women, at least in younger people, than in men. And so in this particular case, we have the production of antibodies for unknown reasons that destroy acetylcholine receptors in the motor end plates. And so we have everything's fine except for this one aspect, and this results in severely reduced muscle contraction as a result of the fact you have the same amount of acetylcholine released, fewer receptors for it to bind to, and so you have this reduction of action potentials that occur on the sarcolemma. There's enough acetylcholine released to generate action potentials initially, but there's a slightly lower level of acetylcholine released in these successive action potentials that arrive to generate contractions, and eventually you have these lower levels of contraction as a result of a deficiency of acetylcholine receptors.